Good evening, everyone, and thank you so much for joining us. In the interest of time, I will keep my introduction very short because we want to carve out as much time as possible for our guest speakers. But I will say that this evening, I think, represents just a great opportunity for people to hear directly from our scientific and research leaders, our clinical leaders, exactly what the Institute is doing. And I think you're going to come away from this meeting very enthusiastic about our mission and optimistic about the impact of our work. So we are very pleased to have with us this evening Dr. Juan Dominguez-Bendala and Dr. Ernesto Bernal-Mizrachi, two leading lights at the Institute, conducting some of the most transformational research that's taking place, not just at the Institute, but in the world of diabetes research in general. So I'm very eager to turn this over to them and have them speak with you. I'd like to introduce them briefly. Dr. Juan Dominguez-Bendala is director of the Pancreatic Regeneration and Stem Cell Lab at the Diabetes Research Institute. He has spent over 2 decades advancing regenerative strategies for T1D. Trained in the United Kingdom, Dr. Bendala's early work included pioneering efforts in human stem cells. His lab is leading global efforts in beta cell regeneration, using live human pancreatic slices to track how insulin-producing cells form in real time. His lab, again, is just really, a leading light in the field. And just last month, his team published a major study in the prestigious Nature Communications journal showing that a small peptide, THR123, can trigger pancreatic regeneration and normalize blood sugar in diabetic models without cell transplants. This breakthrough paves the way for drug-based regeneration therapies, teaching the pancreas to heal itself. Dr. Ernesto Bernal-Mizrachi is chief of the Division of Endocrinology, Diabetes, and Metabolism at the University of Miami Miller School of Medicine, and he is deputy director of signal transduction at the Diabetes Research Institute. Dr. Mizrachi is a highly regarded expert in endocrinology and clinical diabetes research. His work focuses on how pancreatic beta cells function and fail in type 1 diabetes. He's received major research funding from the NIH, the Veterans Administration, and Breakthrough T1D, formerly JDRF, and he's published over 100 scientific papers. His current DRI-funded project explores immunometabolism with the goal of unlocking new drug targets for T1D. Gentlemen, we're so pleased to have you with us this evening. The floor is yours.
Thank you, Michael. I'm going to share the screen, and I hope that you can see my screen now with the invitation. I'm going to put it in slideshow mode. And Ernesto, go
Yes,
ahead.
perfect. Okay. Good evening, everyone. It's my distinguished honor to spend the next 60 minutes with you. And, and I have to start with saying that as a physician and as a scientist, I have a great passion for diabetes and in particular for my favorite microorgan, the pancreatic islets. Next. And I hope that during the presentation today, you will learn about the importance of the islet as a key component for both the development of type 1 and type 2 diabetes, and how a lot of the efforts, not only at the DRI, but in general, are focused on trying to regrow the insulin producing cells as a therapeutic strategy for type 1 and type 2 diabetes. Next. So before we get into that, I just wanted to give you some brief information about what is the background on type 1 diabetes and in type 2 diabetes briefly. But I'd like to start with saying that what happens in type 1 diabetes. So Usually what happens is that your own immune system recognizes your insulin producing cells are foreign cells and mistakenly destroy the beta cells. And that's where what happens when you don't have insulin producing cells, your insulin levels are low, and that's where glucose increases. Usually begins in the childhood, but let me make a parenthesis here. What we initially thought about type 1 diabetes being a disease of a child or young people is not longer 100% true. We see many patients that present at older ages, and they present with diabetes. And if we didn't have the testing for type 1 diabetes, they probably will go treated as type 2 diabetics. And we see that more frequently. And at the same time, we also have seen an increased growth in the type 2 diabetes in the children, especially with the increasing prevalence of obesity in children. And in the slide here, I just wanted to show you, no, before
Yeah, sorry.
this one, yeah, sorry, you can see how basically the process occurs where the immune system starts invading the islet, and with time, these cells enter the islet and at the end destroy them. Next. And here you have a microscopic view of the pancreatic islet. What you can see in the periphery on the pink is the acinar tissue, which is the tissue that produces pancreatic enzymes that will help digestion of the food. And in the center, you will see the pancreatic islet. Now, this pancreatic islet, when in the type 1 diabetes process, gets invaded with the immune cells, and that's the little dots that you see inside the islet. And you can see how those beta cells or the insulin producing cells are shifted and destroyed to a small area of the islet. So that's what happened in type 1 diabetes. Next. Now, how do this happen? So we typically learn about diabetes when the patient presents with very high glucose levels in the hospital with something what we call diabetes ketoacidosis. But in reality, this process has been going for many, many, many years. And as you can see in this slide, uh, this shows how type 1 diabetes develops. And all the individuals with type 1 diabetes are born with a genetic predisposition, and is typically a trigger sometime during life, and we still don't understand what this trigger is. And some of the hypothesis is that some viral infections can trigger this immune attack to the pancreas. But here on the left, you can see in the green what the number of beta cells are, or what we call beta cell mass. And you can see that after the insult or this triggering event, the number of beta cells start decreasing, and people can be asymptomatic for many time until this number of beta cells gets to a critical point that they cannot handle more the glucose, and that's where diabetes develop. Next. Now that was all about type 1 diabetes, but I just have two slides about type 2 diabetes just to highlight the importance of the islet beta cells and the pancreatic islet. And in type 2 diabetes, what happened is that with the increase in nutrition and overnutrition and the Western diet, people start gaining weight, and that obesity progresses. However, we need to know that the majority of the obese individuals are not diabetic and they have insulin resistance. Only a minority of the obese individuals develop type 2 diabetes. And this happens because those islets are getting tired of fighting against the insulin resistance. And this is what is shown here. What you can see here is that as individuals increase in weight, those beta cells start expanding and trying to adapt to the insulin resistance caused by increasing weight and obesity, to the point where the cells get tired and they die. So at the end of type 2 diabetes, there is a decrease in the number of islet beta cells and a decrease in insulin, and that's where these individuals are needed for are treated with insulin. So this is important because you can see that in both diseases there are different stages of interventions and potentially different strategies for the treatment and the cure of both diseases. Next. Now, in terms of the treatment for type 1 diabetes, I've been in the diabetes field for almost 30 years, and I have lived a lot of the developments of the treatment for type 1 diabetes, from initially porcine insulins to human insulin, and after that, all the human insulin analogs, which are human insulin with some changes that made them more fast acting or long lasting. And in addition to that, we now have an increasing the number of tools that are used for the treatment of type 1 diabetes, including insulin pumps. Now, with the glucose sensors and the closed loop systems where the sensor can talk to the pump, this technology has improved the treatment of type 1 diabetes. However, they are still far from being a complete cure of the disease. And that's what next leave us with the next slide. So the next generation of research will try to investigate or identify the different strategies to replenish these beta cells. And in this slide, you can see how science is changing the game and potentially developing new strategies for type 1 diabetes therapy. So in the left, you can see the cell-based insulin delivery, which initially was done with transplantation of islets. And I think that Juan will talk a little bit about the the islet transplantation in the next few slides. And now what we will discuss a little bit later is how we can maybe increase the insulin producing cells, get back to the same number, and that's what we call beta cell regeneration. And this is another strategy that is being investigated to replenish the number of beta cells. And in the next, you can see how turning other cells within the pancreas to beta cells could be a different strategy. And Juan will also be mentioning some of that in the next few slides. In the middle, what you can see is that there are some other therapies that target the treatment of the immune system, which is the second component of type 1 diabetes. And there are now new medications that are approved by the FDA that can train the immune system not to attack those insulin-producing cells as a potential therapy. And finally, the last major breakthrough in research in terms of type 1 diabetes is how you can develop insulin-producing cells from stem cells. And this is a very exciting avenue, but it still has some of the same limitations that the islet transplantation had, which is the use of immunosuppression. And I think that with the theme of islet transplantation, I'm just going to turn over to Juan, so he will expand a little bit more about islet transplantation and some of the cell replacement therapies in his lab.
Thank you, Ernesto. Yeah, I'll take it from here for a few slides. So yeah, beta cell replacement, as it is immune modulation to stop autoimmunity, is one of the major themes of diabetes research. And the DRI has been a pioneer in islet transplantation, which is the first cell therapy for beta cell replacement. And that was pretty much developed by our former scientific director emeritus Camilo Ricordi, whom you can see here to the right, who in the late 80s developed a method that is still used all over the world to isolate islets from a cadaveric donor and transplanting them into recipients with type 1 diabetes. The process, in essence, entails taking the pancreas from a donor, and we can digest the pancreas into the single components. You can separate all the different cell types that you have in there, and then you fish out, so to speak, the islets through centrifugation, and then you put those islets into the liver of a recipient. There are many reasons why we put the islets in the liver as opposed to the pancreas. I don't think that we have the time to go into that, but it seems that they work very well in there. I always show this video. That's the Ricordi chamber, one of the machines that Camilo invented, and there's a human pancreas inside. And what you see there is a process of digestion. At the end of the process, you have a soup that contains all the different cell types of the pancreas. We can separate the islets from that soup. And now we go to the patient. We localize the portal vein like you see there. We put a cannula through the portal vein of the liver, and those are the islets that fall by gravity. The whole process takes about 20 minutes. It's an outpatient procedure, and as you can see, immediately after the transplant, the islets are already functional. Of course, it takes a bit longer for them to revascularize and be fully functional, but it's very fast. So here I can show you, this is a study that was published a couple of years ago that basically reports all the islet transplantation procedures that have been done by the CIT, which is the Clinical Islet Transplantation Consortium, which basically is 12 centers across the world. And this study reports on all the transplants conducted over 10 years by all these centers, right? The good news is in the abstract of the paper is that the results are great. Basically, 75% of the patients who receive an islet transplant are free of insulin long term. However, if you look at the fine print, you can see that they just transplanted 72 patients in total over 10 years in all these centers across the world. If you look to the right, this is the list of authors, and there are nearly twice as many authors of that paper than patients were transplanted. So clearly there's a limitation. There's only so many patients that you can transplant, and this is an example that this is not going to be a cure for type 1 diabetes for as long as we need a source that is not unlimited, and that's the case with islets. So that's why, and Ernesto was mentioning this before, at least islet transplantation has opened the door to the new wave of clinical therapies based on the stem cells. Stem cells, you can multiply them in the lab, and you can get billions of them in a matter of weeks. You can transplant the cells after you differentiate them into whatever you want. You can transplant them into liver cells, differentiate them into liver cells or whatever organ of your choice. Pretty much we have a protocol for every. But beta cells, it took a bit longer, but we can finally get beta cells, and we can transplant these cells. And again, the source is unlimited because we can proliferate or make these cells divide for as long as we need. Now there's interest of big pharma. Vertex is a large pharmaceutical company, and right before the pandemic, they invested nearly $1 billion in these approaches. And they started to do clinical trials right away in the year 2021. And that made headlines all over the world. This gentleman over here is Brian Shelton. He's the first person who received stem cells that were coaxed into becoming beta cells. And Brian had been diabetic for many decades. He was in his 70s. And as you can see here in the bottom, he started with a very poor control, taking a lot of insulin. And after 6 months, he didn't have to take any more insulin, and he was 100% of the time in range. So that was astonishing. The first case of diabetes reversal using stem cells. Unfortunately, the clinical trial was not without hiccups, and last year the clinical, assay had to be stopped temporarily because 2 patients died, including, unfortunately, Brian. Presumably, the reason is that they had to take immune suppression, and when you've had diabetes for a long time and you have many complications, it's not a good combination to have immune suppression. It comes with a lot of side effects. The problem was not the cells that we transplanted, but rather the regimen that we had to use to keep the cells alive within the patient. So the field in general, this was a kind of a proof of concept, but we wanted to put the cells without immune suppression. And for that, we've been trying to put the cells inside a device, an immune isolation device. So that would be, imagine like a protective kind of enclosure where you put the cells and then you transplant that device under the skin, and then the cells are supposed to be protected. There was a clinical trial, and unfortunately, just a few months ago, had to be stopped because the cells didn't survive. The device didn't work as expected. So right now, the clinical trials continue, but it's with immune suppression, which obviously we know is not going to be a long-term solution, but it's something to start testing in the meantime. So in summary, stem cell transplantation is very promising. It's happening. When I joined the DRI 25 years ago, it was a dream, and now it's happening. We are having wave after wave of clinical trials, but we still have a lot of work to do in terms of protecting those cells because the immune isolation devices don't work yet. And obviously, we want to avoid the drugs to prevent immune rejection because they are very dangerous and have many contraindications. So there are other ways to do things, and we're going to get into that. I'm going to pass the screen now to Ernesto and the concept of regrowing the cells within the pancreas without the need for transplantation. That's what we call regeneration. Ernesto.
Thank you, Juan. I think that as you could hear from Juan, this strategy that he mentioned is basically replacing the beta cells or growing beta cells from stem cells. In this case, is what is regrowing means that you will make your own beta cells duplicate and replicate to a manner that you can achieve a normal beta cell mass at the end. And this slide is just to tell you some very important concept that many people don't know. As you can see here on the left, this is the beta cell mass or the number of beta cells that you have in the pancreas. And incidentally, the human islets are about only 4 or 5% of the total mass of the pancreas, which is between 70 to 140 grams. And what you can see here in this slide is that you get to the maximum beta cell mass by 20 years of age, and that's the maximum beta cell that you will have, and you maintain that number of beta cells throughout your life. So you can understand now how when you have type 1 diabetes and your immune system is destroying these cells are not continuously dividing and regenerating. So the cells that you will lose are not able to regenerate. And the little dotted line that you see on the top is what you will see in patients with type 2 diabetes. As you can see, in type 2 diabetes, the insulin resistant made your own cells to grow a little bit, but that's what we call adaptation to insulin resistance. But one thing that is important to know is that the adaptation to insulin resistance is very limited, and that's what at the end is not enough to prevent type 2 diabetes. And in the case of type 1 diabetes, the loss of those beta cells are basically impossible to regenerate from the own endogenous pancreas. And that's how next there are some other strategies that have been investigated and how we can develop agents that could induce your replication or multiplication of your own beta cells. And on the right, you can see the only one agent that has been shown some exciting results with beta cell regeneration, and it's called harmine. And this is just a name that has come from many years, and it's a substance that was isolated from some plants in South America. And there are some researchers at Mount Sinai that have been developing analogs that are similar to this medication for the treatment of type 1 diabetes. Now, one thing I have to say is that the DRI is also participating in these developments and also trying to develop clinical trials to use some of the harmine analogs in humans. Now, the other medication that you can see here is the GLP-1 analogs. These are the medications that you all hear all the time on TV and in radio, the semaglutide, and all these other GLP-1 receptor analogs. So at the beginning, there was a lot of excitement about this medication as agents that could induce beta cell replication and multiplication. But it seems like this was only occurring in mice. But when the human or human islets are exposed to GLP-1 analogs, the capacity of these agents to induce beta cell replication is very limited. Next. So one of the strategies that we have been using in the lab is one compound that was developed by a Nobel Prize. You can see in the middle, Dr. Andrew Shalley. Dr. Andrew Shalley was at the University of Miami until last year when he passed away, and he was the recipient of the Nobel Prize in 1977 with Roger Guillemin and Rosalind Yalow. We just were discussing the contribution of Rosalind Yalow was to develop the technique that is used to measure hormones, and that's how they were able to make some of the discoveries. But he made some analogous to one endogenous hormones called GHRH, And we have been using these analogs to try to prevent beta cell death as another strategy to maintain your beta cell mass. So we can see here that you can only increase the multiplication, but also if you can find agents that prevent the beta cell death, that could be an alternative strategy. And in the middle, what you can see is that when we treated the human islets with this compound in a medium that contains, all the cytokines that are seen when the immune cells invade the islet. What you could see is that treatment of these human islets with this compound really decreased the beta cell death, as you can see comparing the blue and the red. And when we use the same compound in a mouse model that is similar to type 1 diabetes, where we kill all the beta cells with a very toxic agent, you can see in the red line that the treatment with this compound really ameliorated the high glucose induced by destruction of the beta cells, suggesting that this compound was preventing the beta cell death induced by this toxin. And we're now trying to go back one.
Sorry.
We're now trying to use this compound in other preclinical models and perhaps use that in some human clinical trials. The important point is there is already an FDA-approved medication that acts in the similar pathway and that has been used for another type of disease. So that is encouraging because this is a proof of concept and is telling us that this medication is not toxic for humans. Next. And the other interesting experiment that we're doing in the lab is how a missing protein that we call here 4E, be simple, can protect the mice from diabetes. And this is a model that is very similar to type 1 diabetes in humans, and it's called the non-obese diabetes model, or NOD. And you can see in the red the animals that lack this gene or protein, and you can see how they are 100% protected from beta cells. So with funding and research support from the DRI Foundation, we're trying to understand how not having this protein could be causing the protection for type 1 diabetes. And only that, maybe we can develop pharmacological agents that can mimic this response. And I think this is very exciting because we think that this molecule can not only protect the beta cells and also avoid that they die under an immune attack, but also they can modulate the immune system to prevent the attacks over the beta cell. Next. And then moving along, next. The animation. So I have shown you about the proliferation and this, what is called DRI inhibitor, is the harming molecule that I told you. Another strategy that has been explored is how we can transform the other cells in the islet, like alpha cells, into beta cells. And that's another exciting strategy that has been investigated in some labs, including my lab. Next. And then I have mentioned about the GLP-1 receptor analogs, the semaglutide, tirzepatide that everybody hears in the commercials. They initially were described as an insulin or a regenerating agents, but at the end we don't see this in humans. And the last strategy that is important to mention, because I'm going to turn this over to Juan again, is how we can transform only the ductal cells, which is the other cells in the pancreas, to become beta cells. And I will just turn this over to you, Juan.
Thank you, Ernesto. All right. So, yeah, we're going to talk about the ducts, and that's something that people don't think about normally when we talk about the pancreas. But when you look at these ducts, basically what they do is to conduct the digestive juices to the duodenum. So it's a very pedestrian function if you think about that. But it's very important, and they're all over the place. If you look at the cast of the pancreas, the ducts are pretty much everywhere. And during the embryonic development, they are the ones that give rise to entire new regions of the pancreas, including new islets. The idea that the ducts can give rise to new islets, it's not mine, it's not ours, it's not actually new. It's the first one who came up with the idea was this gentleman over here whose name is Robert Bensley in 1911, more than 100 years ago. And he was painstakingly dissecting all the pancreata from guinea pigs. And he's the first one who noticed that all the islets were in close connection to the ducts, as if sprouting from them. As you can see, he was the first one who said in very clear words, they are forming from these glands, from these ductal, structures. And it makes perfect sense if you think about that, because that's exactly what happens, as I mentioned before, during embryonic development. When the pancreas is forming, it is the ducts that branch out and give rise to new islets. And that is actually carried over to the newborns. When you are born during the perinatal period, even though there's a lot of expansion of beta cells by proliferation of pre-existing beta cells, there's also a lot of neogenesis, as we call it, ductal cells becoming islets. If you see here to the left, these two pictures that I got from our friend and colleague Sarah Richardson from the University of Exeter in the UK. And you can see lots of new islets in teal color sprouting from these ducts. And that's something that happens during the whole perinatal period. Now, it's true that in adult life, if you have a healthy pancreas, there's no reason for you to have regeneration because, in fact, beta cells are very long-lived. They can live 20 years if nothing wrong happens to them. And if there's any death that needs to be addressed, another beta cell will replicate and take the place of the cell that died. But what we and others contend is that when the pancreas is under stress, the likes of pancreatitis, autoimmunity, like it happens in type 1 diabetes, or even in injury of the pancreas, there's a partial reactivation of the embryonic program, and the ducts start to form new islets. You can see this even in new onset type 1D cases, like this one to the left. You can see the ducts, and they're new islets that are trying to sprout and form. Ernesto said it very well before. This is a natural reaction of the pancreas. The pancreas is trying to compensate and regenerate, but it's never enough. We know how that story ends. The pancreas is trying, but it doesn't get much, much farther than this. So we are aiming at harnessing this process and boosting it, having, tried to stimulate it from a pharmacological perspective and make it happen faster and more extensively, because nature can only get so far. So we have spent the last 10 years or so in my team, together with Ricardo Pastori, a colleague of mine at the DRI, investigating what these cells do, where they are. And just to give you an idea, because an image is better than anything I can tell you, they are hiding in plain sight within the pancreatic ducts. Those cells that you see there in red, those are the pancreatic progenitors, the stem cells in the pancreas that can give rise to new islets. And I say that they're hiding in plain sight because they're intermingled with all the green cells that you see there that are the normal ductal cells. So this is a region that has a lot of them, by the way. Just to give you an idea of how powerful these cells are, we can extract them from the rest of the ductal tissue. We can purify them. And then we can use a model by which we can transplant these cells under the kidney capsule of immune deficient mice. So these are mice that don't have a functional immune system, and therefore you can transplant human cells into them and they won't be rejected. So we transplant these human pancreatic progenitors that we found in the ducts into the kidney of these mice, and then we wait a few weeks. After just a couple of weeks, you can see human insulin being produced in the blood of these mice. But when we look at the kidney, we can see something like this. It's a mini human pancreas. So the cells that we transplanted, even though they were single cells and there were only a handful of them, somehow they have the memory or the instructions, the blueprint for creating a brand new pancreas. And this is a human pancreas is making human insulin. You can see there a little islet in white, but you also see the acinar tissue that's making digestive juices. You can see the ducts. It's amazing. So that's how powerful these cells are. When we are trying to reawaken these cells, we are tapping into a very primitive regeneration mechanism, something that happened during very early embryonic development. So we have that potential. The question is whether we can activate these cells in a way that will lead to the formation of new beta cells within the pancreas without the need for transplantation. Well, we found, Ricardo Pastori and I found a molecule called BMP7, which is a growth factor. It's a naturally occurring growth factor. It's in your blood and it's in mine. It's made in the kidneys, by the way, but it's also found in the bone. And it's fuel for stem cells. Every time you have stem cells and you have BMP7, which is one of the many BMPs out there, you have proliferation of these cells. And when you remove the BMP7, they stop dividing and they become whatever tissue they are in. I mean, if it's the liver, they will become liver cells. If it is the pancreas, they will become new islets. The good thing about the BMP7 is that it's already being used. It's been in use in the clinical practice for the last 2 decades. Every time you have a spinal surgery and they need to fuse vertebrae, they use this glue, which is made of BMP7. What it does is to activate the progenitor cells within the bone, and it regrows the bone. We found that BMP7 also activates these progenitor cells in the pancreas and induces the formation of new beta cells. So the question that we had, and this is the paper that we just published a few weeks ago, at the beginning of July, is whether BMPs given pharmacologically to an animal model of diabetes, if it can stimulate the formation of new beta cells without the need for transplantation in the pancreas, where nature intended for islets to grow. So I'm going to go through this very fast because, this is not a scientific presentation, but I just want to tell you that we use this small molecule or peptide called THR123. To all practical effects, think of it like as BMP7. It's a smaller version of BMP7. BMP7 is a large protein. It's difficult to synthesize. It's very expensive. THR123 is a smaller version of the protein, and a family member of THR123 has already been used in clinical trials. It works exactly like BMP7. So this is the key graphic of the paper. If you look at the blue line, those are diabetic animals. We can make diabetic mice, and the sugar levels are over 600. That's very diabetic. As you can see at the bottom, the red line, those are animals that we treated with THR123, and the sugar levels are around 200. You may say that 200 is still very high, but not for mice. Anything below 250 mice is normoglycemic. So as you can see, there's a huge separation and a huge advantage of using this THR123. But what's happening for this to happen in the pancreas of the mice? So let me guide you through what we saw. This is the pancreas of a normal mouse, and you can see in red insulin-producing cells. So what you see there is the islets of a normal pancreas. This is a cross-section of a pancreas in which stain for the cells that make insulin. When you look at the pancreas of a diabetic mouse, this is what you see. There's no red there. There's no islets. We kill all the islets when we make these mice diabetic, and there's no beta cells. Now, if we use a diabetic mouse and we treat it for a month with THR123, what we see in the pancreas is this. There's a multitude of smaller islets. They are seemingly sprouting from the ducts, the very regions where we had identified the stem cells, the progenitor cells, and they're all over the place. How do we know that these are new beta cells and perhaps not cells that survived the attack of the chemical that we use to make the animals diabetic? Well, we know that they are new because we use something called BRDU. Let me guide you through what it is very quickly. Every time a cell divides, when a cell forms, it's because it's coming from a parent cell, right? The cell divides. When the cell divides, you have to create a copy of your DNA. And that happens in this graphic. You can see the original strand of DNA. You separate the two strands, and then you make a copy using the building blocks that are in the environment. These building blocks are the nucleotides. But we can kind of trick the system because we can add a fake nucleotide, a fake building block, which is called BRDU. This is something that incorporates into the new DNA, and we can label it in any color that we want. We label it in green, meaning to all practical effects, when you look at the cells after BRDU administration, if you have a cell and BRDU is in the culture medium or in the mouse, if we give the BRDU to the mice in the drinking water, any daughter cell, any brand new cell that you have in there will be green. All right, so with this strategy, what we did was to treat the animals, diabetic animals, with THR123, but we also gave them BRDU in the drinking water, meaning that if we have formation of new beta cells, they should be green because they are the result of the division of pre-existing cells. All right. So let me give you an example of what a control looks like. This is a normal non-diabetic mouse, and we are looking at the pancreas, and that's a beautiful islet that you see there in white. You can also change the channel and look at the green color. The green labels cells that are dividing or that have divided during the treatment. In this case, because there was no treatment, this is a normal mouse, there's no BRDO there. There's no signal whatsoever. And it shouldn't, because there's a normal islet, there's no need for regeneration, so there's no new beta cells in there. Now, let's look at the pancreas of a mouse that has been made diabetic and then treated with saline solution, which is basically a placebo, for 30 days. And as you can see, here in the lower quadrant, there's a little bit of insulin, but not much. These are not islets. These are only, I count 1, 2, 3, 4, or 5 insulin-producing cells. There's a lot of BRDU in the other channel, meaning that the pancreas is trying to regenerate. There's a duct in there, and there's a region of active regeneration, but it's not taking the regeneration very far. That's what I told you before. You're not giving anything to boost the process. The pancreas is trying, but it's not getting very far. Let's look at the pancreas of a mouse that was made diabetic and was treated with THR 123 for 30 days. You can see islets like that one, fully formed, beautiful, with all the cells, all the beta cells that you see there in green, meaning that each one of those dots that you see there in green is a brand new beta cell that wasn't there before. It's BRDU positive. So we think this is tremendously exciting. It's something that we gave to the mice, an injectable chemical that regrows the beta cells. And these cells are functional because they decrease the hyperglycemia that they had in the first place. So we are very excited, but we always have our friend and colleague Jay Schuyler here at the DRI, who tempers a little bit our enthusiasm, and he's always reminding us something that's very true, that we've been able to cure diabetes in mice in hundreds of different ways, and none of them has ever worked in humans. So we had to take the results that we have in mice with a grain of salt. But fortunately, we have something in our favor. This is not the only model that we've been working on. We have another model which is human-based, and it's the use of pancreatic slices. It is exactly what it sounds like, and what you're looking at in that little video that you see there, these are live slices of a human pancreas. So when we have a donor pancreas, when the organ is still alive, we can section it, and we can keep those slices alive in the lab for up to 2 weeks. That's something that not many people can do, but we can, thanks to a technology that we developed in the DRI, together with another scientist and a colleague of ours, Chris Fraker, that provides enhanced oxygen to the slices, and we can keep them alive for 2 weeks. And those are 2 weeks during which we have a window into a real living, breathing human pancreas where you can see everything in there. You have the acinar tissue that makes all the digestive juices. You have the ducts where you have the stem cells, and you have the islets. It's, again, the whole ecosystem of the organ. All right, so we can do something else. We can get slices from type 1D donors. So these are people who had type 1 diabetes at the time of their passing. And thanks to an organization called MPOL, which distributes these tissues nationwide, we have access to these tissues, very valuable and precious tissues from people who had type 1 diabetes when they passed away. We have a strategy by which we can infect these slices with viruses that act like Trojan horses. What they do is to deliver specific markers to the cells that are in there, and we can label them in different colors. For instance, the ductal cells, where we have the progenitor cells, the stem cells, they will be tagged in red. The beta cells, if you have any pre-existing beta cells, they will be tagged in blue. And if you have any transition from a ductal cell to a new beta cell, there will be an intermediate stage where the cells have both colors, the red and the blue, and you can see them in magenta or purple. So look at this. We put everything into a microfluidic chip. This is a fashionable concept in biology, which is to put an organ in a chip. This is something that we did also in collaboration with another scientist who is affiliated to the DRI at the University of Miami, Ashu Agarwal. And we have total control over everything that goes in or out of that chip. And we have a human pancreas section in there. So let's focus on a region of that pancreatic slice that you can see here in this rather busy slide. But you can see here that we treat with THR 1, 2, 3, 4, 5 days. And then we look at live cells in different colors. This is as we look at them under the microscope, we can see cells in different colors. The top row, you can see that there's more blue as time goes by. There are new blue cells that are being formed after you add THR 1, 2, 3. That means that those are new beta cells. And every time there's a new beta cell there, a new blue cell, it appears through a transitional magenta stage, as you can see here at the bottom. You can see even cells at the wall of that duct that you see at the bottom. They are transitioning in real time. You can see a red cell transitioning into a blue cell through an intermediate magenta stage. So that's biology happening in front of your very eyes in real time. Not only are they transitioning and becoming beta cells, we know that they are functional because when we test whether they produce insulin in response to glucose stimulation, they do. They become brighter with another dye that we have in there, meaning that they are producing insulin. This is the very first time that we can show that we can regenerate functional beta cells in a section of the human pancreas of a patient that had type 1 diabetes. And I cannot stress enough the importance of this because we can do as Skyler mentions many times, we can do all kinds of things in mice and we can cure them left and right. But to show functional beta cell regeneration in a slice of a human pancreas from a type 1D donor, that's something that we think it's a major breakthrough. So that's why we are confident that this approach that we have tested in mice will have an application in humans. This is another example of the cells transition, as I mentioned before. What we are doing now, the next steps, because obviously that's a question that everybody has, we had to do now a lot of boring studies. The exciting part is over. We have shown that this works, but now we have to convince the FDA that this is safe and that we can reproduce it. So now we have to do lots of experiments to test, ensure the reproducibility of these results, to ensure the safety. This is super important. And this is something very interesting because many people are concerned that we are putting a growth factor. Well, the growth factor we are injecting, so it's going everywhere. It's not going only to the pancreas, but we see the effect only in the pancreas. We don't see anything else in the liver or in the muscle or in the brain. Why is that? Well, BMP, it's like I mentioned before, fuel for stem cells, and stem cells are residing in all the tissues and they contribute to the regeneration of those tissues. When you have injury in a tissue, and that's what happens in the pancreas when you have type 1 diabetes, that's where the BMPs are working. In fact, they upregulate the expression of the receptors for BMP7s and the THR molecules because it's like an SOS signal, like help us, we need regeneration. That's where BMP7 binds. And we see that happening in the pancreas because that's where the injury is. It's not in the liver and it's not in the muscle, it's there. So we think it's very specific to where the injury is, and that's what happens in type 1 diabetes. Our plan, and this is funded by obviously our foundation and Breakthrough Type 1D, our plan is to talk to the FDA in 3 to 5 years and show them all the data, showing the safety and reproducibility of these findings and take it to clinical trials. So I'll stop there because otherwise I can't keep talking, but Ernesto will give you the main conclusions of everything that we've been discussing so far. Ernesto.
Thank you, Juan. I guess in the interest of time, I'm going to highlight the major conclusions. And there are shown some of them here. I think we have made significant advances in the treatment of type 1 diabetes. However, the cure is still elusive, and the support of the foundation will be instrumental to develop some of the new avenues for the cure that you saw today, including the exciting results from Juan. And the findings today were just some of the ongoing research that we have at the DRI, trying to find a cure for type 1 diabetes. Next. And I have to say here that emphasize the fact that at this point, in moving to the future, we're going to need combination therapy, so agents that will ameliorate the immune attack to the islet, and then other agents that will induce not only maybe beta cell expansion, but also beta cell neogenesis. And with that, I think that would be one of the future strategies using different compounds or cocktail of compounds that can be used to treat and to cure type 1 diabetes. Next. And finally, I want to focus on the last conclusion. While a cure for type 1 diabetes remains a goal, we're making rapid and meaningful strides toward that vision, fueled by research, innovation, and hope. And as the key point towards the future is research is what's going to take us to the cure of type 1 diabetes. And I guess with that, I close my conclusions, and we can open this to the debate. But before that, I would like to acknowledge all the members of my lab. Some of the ones are shown here. I also like to show Dr. Andrew Shalley, who, in honor of him, he passed away last year, and all the funding agencies, not only including the VA and Breakthrough, but also Yusuf Line, the DRI, and the DRI Foundation. And then I will turn this over to Juan, who has his acknowledgement.
Yeah, I had another slide of acknowledgements, which is very long because this is obviously the teamwork. We are simply presenting the work of many others over the years, but I'll stop there and stop sharing the presentation and open the floor for questions. And yeah.
Thanks so much, gentlemen. That was tremendous. We have a number of questions that have come in, and if I don't get to your questions, I assure you we will follow up individually following the call, but thank you for your submissions. First, can you speak at all to the progress from Sana Biotech that was announced in the New England Journal of Medicine about clinical data from the transplantation without immunosuppression?
Yes.
You want to take that one?
That's fine. Yeah, this paper was just published in the New England Journal, and it's from a group from Uppsala in Sweden. And basically, what they have done, I mean, during the conversation today, we showed that you could maybe generate beta cells from stem cells, but we also mentioned that they had the drawback that they will still be recognized as foreign and they will be attacked by the immune system from the recipient or individual. So what the researchers have done in these studies that they created cells by a technique that is called CRISPR-Cas, and these cells are not being able to recognize by the immune system, and they were transplanted into a muscle of one individual. So I think that this is exciting, and this is where the field is moving, trying to not even generate cells, but also cells that won't be recognized by the immune system. So
Yeah.
one comment.
That's absolutely right, and I agree. This is where the field is moving. I just have a word of caution for that. When you make stem cells invisible to the immune system, there's a risk that is common to all pluripotent stem cells, which is that if some cells do not completely differentiate, they may remain active and proliferating after they are within the host, and they may form a tumor called teratoma. That was very common 10 years ago and not so common today when we transplant these cells into mice. But before, about 20% of the mice that got these tumors, they're not malignant tumors. They grow in place and you can remove them. But it was something obviously that you don't want. If you are getting treatment for type 1 diabetes, you don't want a tumor. So my concern is that if now we're going to make stem cells invisible to the immune system and a tumor is to form, there's no way that the immune system can take care of that tumor. So far, if you have immune suppression, you simply have to remove the immune suppression and the cells are attacked and die if you had a tumor. But if you have invisible cells, I think that other measures are necessary. And my team has been investigating a little bit on this, and we developed an approach by which we can make stem cells safer. If they were to form a tumor, we can activate suicide genes that will kill those cells and those cells only, the ones that give rise to a tumor. So those are safeguards that I think are necessary if we are going to move in this direction. That study, by the way, was done in islets, if I'm not mistaken, but the idea was to move it to stem cells, and that's why I expressed my concern. But in islets, you don't have that concern.
Yeah, exactly.
Yeah.
Yeah, those were islets, so there is less concern about the, pluripotential of
Right.
But by the way, we will be posting in the chat a phone number for anyone who wants more information directly on our clinical trials. Please use that phone number to reach out to the DRI directly. Next question. Most of us, gentlemen, have been hearing about a diabetes cure for years, which makes it difficult not to be somewhat skeptical. What makes this moment in beta cell regeneration different? And what milestones would you need to hit in the next 2 to 3 years to feel that beta cell regeneration is ready for broader clinical trial?
Okay. Well, I think I alluded to that in one of my last slides. The plan is there to make sure that the approach that we are pursuing is safe and is reproducible. And we have a few things in our favor. One of them is the fact that BMP7, the fuel that we talk about, has been used for 20 years already in the clinical practice, as I mentioned, for bone fusion. And there's another sister molecule of THR-123 called THR-184 that has also been used for another irrelevant condition, something that has nothing to do with diabetes, but has been going through phase 2 clinical trials in hundreds of patients. It was given systemically, and no side effects were reported. So we are confident that, this is not a chemical agent that is not in nature. It's something that is in your body. We're simply increasing the dose and making it work where it is needed. But going to the first part of your question as to, why do we think that this is special and not just one more? I don't think that I've said anywhere, and I agree, I'm sure that Ernesto agrees with me, we are not claiming that we have a cure. This is the beginning. Some people are telling me, congratulations, that's a wonderful. This is only the beginning of a path to the clinic, and many things die in the process. We hope it's not going to be the case, and we have another advantage in here, which is the fact that it's not working only in mice; it's working in type 1D sections of the pancreas. I mean, sections of the pancreas of type 1D donors, meaning that it doesn't get any closer than that to a real clinical situation. To look at the pancreas of a patient who had type 1 diabetes and see new beta cells growing there and making insulin in response to glucose stimulation. So there are many things that are aligning in our research that makes us optimistic, but sometimes the hype it's not our fault. It's the way this is relayed or conveyed to the media, and you need the headlines, right? And sometimes things are oversold. We are very cautious. We are not claiming that this is a cure, but we think it's a very important step in the right direction. It doesn't come out of nowhere. We've been working on this for 10 years. And this is the result of 10 years of work where we've been going stepwise through the whole thing. And I think this is the way to go, little by little. Breakthroughs are simply, moments in which you see the horizon closer and not just the cure. This is my personal opinion. Ernesto may or may not agree with this, but we put so much weight on the word cure. That's a lot of pressure, and we are seeing as we speak brand new therapies that are changing the way that we manage the disease. Look at how diabetes was treated just 10 or 15 years ago and how it is treated today. And even, I'm not talking about the management of the disease, which is light years away from what it was 10 years ago. There's now an agent, a pharmacological agent in the market, the T-Sealed, which delays the onset of diabetes in people with genetic predisposition by 2 years. And that's a major breakthrough. I mean, it may not sound like much to you, but 2 years is 2 years. Perhaps the next wave of clinical trials will be 4 years. And who knows if we can stop the progression of the disease during the honeymoon period in the next few clinical trials. So it's a stepwise process. And my hope is that by the time the cure comes, people may not even notice, perhaps, because there have been so many advances in the field that there are therapies that are very effective already and that lead to a complete change in the quality of life of these patients even before the cure arrives.
So I just want to mention, Juan mentioned some important points. I've been doing clinical diabetes and research for 30 years. And when I was in my fellowship, we were using insulin, and insulin pumps were still very rudimentary. And I have lived the evolution from that to the islet transplant, which was a major breakthrough, and the DRI was a major player on that. And then moving that, now we're moving to a different era where we are building all the elements that are needed for a cure, meaning develop agents that will modulate the immune system. And now these other strategies that will not only increase beta cell regeneration, but also formation like your compound and others. And you see all the New England Journal study now here. So I think that the excitement is that there are now a lot of elements in place to maybe design very exciting strategies for the future.
Thank you. Thank you both. You touched on this earlier, but do you think a biological cure will require combining beta cell generation with immune modulation, or could regeneration alone potentially work without immune protection?
Well, yeah, that's a really good question. And the short answer is that we don't know, and it probably will require combination therapies, as Ernesto pointed out before. However, and this is perhaps a long shot, but there's a growing realization that type 1 diabetes, even though it's an autoimmune disease, there's also a beta cell component to the disease. It seems that obviously when you have genetic predisposition to autoimmunity, you have what we call a trigger-happy immune system. And the immune system is very prone to overreact, right? But why does it attack the beta cells and nothing else in people who develop type 1 diabetes? And when we look at the etiology of the disease and how it progresses, sometimes the beta cells are sick way before the attack of the immune system. And they are sick and they are not producing as much insulin, and they are making all these aberrant proteins that are not found in nature. In a way, they're provoking, taunting the immune system. And when you have a very, trigger happy immune system, that's a very deadly combination, so to speak. So the idea that the beta cell is sick in the first place, it's gaining traction. And our approach, if you think about it, if we are now regenerating brand new beta cells that are like the ones that you find during embryonic development, that don't have any of the sickness, any of the abnormalities, any of the aberrant proteins that the ones that the immune system recognized at the onset of the disease, who knows? Perhaps if we depress the immune system temporarily and allow these cells to regrow, when we put the immune system back online, they won't be recognized as the ones that offended the immune system in the first place because they are different, they are not sick. So we are investigating this as we speak. We don't know. And I'm not going to put my money on this because in no likelihood we're going to need some kind of immune therapy along regeneration. But who knows? Something that we still need to find out.
You mentioned earlier, I believe, converting alpha cells into beta cells. Are you concerned about losing glucagon control as a result?
Yeah, so this is an interesting concept. I think the transforming alpha cells to beta cells is behind from all these other strategies. But the nice thing is similar to what Juan mentioned, is that you will build your own beta cells, and that will be an advantage, right? From endogenous cells. Now, what is the importance of glucagon? I think that glucagon is already altered in type 1 diabetes. The function of the glucagon responses is different in type 1 diabetes, and that's why the recurrent hypoglycemia in patients can be very detrimental in terms of quality of life, but also, significant impact for patients. Now, what happened is that in these patients in type 1 diabetes, glucagon is not working and is not able to be the first line response to low glucose. So they already are dysfunctional, and not having glucagon at that point maybe won't have a major impact in the patient, right? Because the function is already impaired. But we'll see. These are still early, and we're still learning a lot of more research in terms of alpha cell biology, which are glucagon producing cells. And this is now shifting, diabetes not to be a unihormonal disease, but also another defect, a second defect from glucagon too.
Thank you. Another question. Many families are focused on where their loved ones might fit into future trials. Is there a patient population in particular that you think could benefit from these therapies first? Are we talking about newly diagnosed patients or possibly those living with T1D for years?
I don't know, maybe I can say something. You saw the nice curve with what happened in the natural history of the type 1, where you have beta cells, and then you at the very end, when you develop the disease, you have very few left. So I think that the initial therapy would be better for the patients that still have beta cells, and that's still very challenging. But because if you're going to regenerate beta cells, you still need to have some endogenous beta cells that you can regenerate or multiply. In the case of Juan's strategy, you can start at any point, because if you have the ductal cells providing beta cells, you can start from when you don't have beta cells, and they will just replenish some beta cells that you lose. So I would say that the higher the chances are for patients that still have beta cells with the current therapies. But the field is evolving so fast that there's going to be maybe opportunities for patients that already are at the later stages when they don't have beta cells.
I agree with that. And you pointed out that if you have an approach like ours, where we are creating new beta cells, it can happen also later in the course of the disease. And in fact, we published a few years ago that these progenitor cells that we have identified persist regardless of the duration of the disease, and you can find them in people who have been diabetic for decades. So there's another case to be made about trying to do this later on. And I completely agree that it makes perfect sense to try at the beginning when you still have residual beta cells, to try to do anything in your power to preserve those and perhaps to expand them. On the other hand, you may also look at the later phases as, when all the destruction has taken place already and there's not so much inflammation going on and not so much an active immune response. Because you know that, for instance, autoantibodies decrease over the course of the disease. Sometimes you don't find them later in life because the immune system has gone back to sleep because there's nothing left to destroy. Perhaps that's a time for us to regrow new beta cells in the absence of any inflammation or immune attack, and then see what happens. So we don't know. For sure, when it comes to trying these approaches that we have developed, the fact that we need perhaps an immune therapy to prevent the death of the newly formed beta cells, that makes us think about the initial cohort of patients that we want to test. So for instance, one example, if we wanted to test if THR-123 works, we may want to go for insulin-dependent type 2 patients who need already replacement of beta cells because there's no autoimmunity going on in there, and we can see if it works, even though our target ultimate target is going to be type 1D. But we don't know because type 2D also has the component of insulin resistance. So there are many, many things that we need to discuss with the people who really know how to take this to the clinic, like Ernesto and our experts in regulatory affairs. They know what needs to be done and what cohorts are going to be the best ones. It's going to be a stage-wise process, and probably, I don't know if type 1D is going to be the first cohort of patients, but we still have to see.
And I just wanted to add, in terms of these agents that I showed that induce the multiplication of beta cells, so those in particular will need, some beta cells left. I mean,
Right.
specifically for those, you will need some beta cells to induce the multiplication and replication.
Right.
A quick follow-up question that came into this point. Will there be different interventions for different stages of T1D, recent onset versus long-term?
Ernesto.
I think that, as I mentioned, the clinical trial landscape is evolving and is trying to use all the elements, and they will be used strategically depending on what stage you are. And I think that the key point is that the trial net, which is one of the biggest type 1 diabetes trial, has really discovered a lot of the natural history of the disease. So what happened from the beginning to the end, and now we can apply that knowledge to design more strategically specific clinical trials according to the tools that we have.
Great. We have one more question, and I think this is an interesting summary question. You are working at the intersection of hope and science. How do you personally balance ambition with the caution that clinical research demands? In other words, help us understand the emotional and professional tension researchers face in pushing forward innovation responsibly while being aware of the community's high hopes.
Good question, right?
You take that first.
It's a balancing act. At the end of the day, we are scientists. We have to get our grants, and obviously we try to push for what we are doing, and try to portray it in a way that is perceived as promising, which we think it is. But I understand that there's a fine line in there, and we have to be also cautious. And I think I do that. I showed today a slide with our friend Jay Schuyler saying this, which is absolutely true. I mean, be cautious of everything that you show in mice, because how many times we have claimed that something is a cure and it didn't work when you try to translate that to humans, right? So we take everything with a grain of salt. And when people congratulate me on this paper or the team, it's like, it's only the beginning. I mean, congratulate me when we are doing clinical trials and they are working. It's a process. I try, at the same time, it's important to give hope. And we get visits all the time when you guys at the foundation tour our labs and we see the faces of the children for whom we are doing this. It's a lot of responsibility for us, but we have to give them hope without giving them the hope that it's going to happen tomorrow, because nothing happens overnight. I agree with something that Ernesto said before, and the word that comes to mind is critical mass. I mean, I think that we are reaching a critical mass of knowledge through a process of trial and error many times. When I joined the DRI 25 years ago, with the stem cells, we were going in every possible direction, and it's only through time and trial and error that we are finding the best promising avenues, and we are sticking to those. And now it's only one or two things. Before it was 20. So that process of elimination and the acquisition of a critical mass of knowledge, that's what makes me optimistic that we are getting closer. Anybody can say we're going to have a cure in 10 years. That's meaningless, but it starts to get some meaning when you've been working on the same thing for 20, 30 years, and you see how things are taking shape. And I think that things are taking shape, and that makes me optimistic. It's not going to happen overnight, and you have to be absolutely certain that what you do can be reproduced and is safe. And sometimes these things take a long time, and for a good reason. And the FDA, we try to think of many times we think of the FDA as the bad guy who is putting brakes on science, but ultimately they are trying to protect the people, and to rush something is often not a good idea. And there's a process, and we have to go through the whole process and go through the steps and hope that it will get there sooner rather than later. I'm very optimistic again that we are reaching that point where everything is converging. Advances in immunological side of the disease, we know now what arms of the immune system are involved in autoimmunity. Before it was all about immune suppression, but we have an immune system for a reason. You don't want to depress the immune system. You want to tinker with it, and now we have the ability to do so. And at the same time, we have a lot of knowledge that has been developing over the years on how to regrow the beta cells, on how to transplant new beta cells. Things are converging to a point that I think is very promising. I cannot tell you how many years, because that would be irresponsible, but it's much, much closer than it was 10 years ago. Ernesto, I don't know if you want to add something.
I just want to compliment that when I get these type of questions, my answer is I let the data guide us, and data generated with very, rigorous research. I think I am privileged in the way that I see patients asking me these questions, but at the same time, I know very well what is happening in research, and I could tell them a balanced opinion based on the data and try to center them more through what is, the truth and not the hype in diabetes research. So the data is key, and for data we need research. So that's what I answer usually.
On that note, I will personally thank you both. And for anyone whose questions weren't answered, we will follow up. And please don't hesitate to email me, mmyers@drif.org. It'll be in the chat with any additional questions for our presenters. Gentlemen, I can only tell you that the comments that are coming in are so appreciative, not only of your insights, but of your candor and your passion. So I thank you. I will turn it over to Michael Burton to close.
Thank you, Michael. Briefly, I don't think there's any substitute for hearing about innovative science directly from the people who are carrying that science out. And that is really the rationale behind the Meet a Scientist series that we hold. And I want to thank you, Dr. Mizrachi, Dr. Bendala, for joining us this evening. I thought it was an outstanding presentation. And as Michael said, the comments are rolling in. The reviews are strong. So thank you. Thank you for everything that you do. I also want to thank, of course, our guests this evening, everyone who joined us. Thank you for joining us. I see some very familiar names, some people who are stalwarts of this organization. I see other new names, folks that I don't recognize, and that's very encouraging. I would like to kind of make this appeal. We can't do this without you. We need your help and your support, and you can provide that in lots of different ways. You can tell a friend about our mission. You can come to one of our many events in the course of a year. You can support us, of course, financially, and that is even more important now, given all the ambiguities of federal funding in recent months. Private philanthropic support is absolutely vital to continue the work that Dr. Mizrachi and Dr. Bendala are doing, and of course their colleagues across the Institute. So we thank our current supporters, we thank our future supporters, we thank you for just being here this evening and for sharing in our mission. I have no doubt that one day we will defeat this disease, and when we do it, we will defeat it together. So thank you so much for joining us, and have a very good evening.